All right, so we are continuing on our study of botany. And so the next part is taking a look at an actual tree and looking at the woody tissue that makes up the bark and what actually makes trees different than any other type of plant. So we've been talking about mainly herbaceous tissues, which are the green fleshy part of plants. We looked at leaves already, whether they're simple, compound, or pinnate, palmate. And so we're just kind of continuing on looking at different types of parts of the trees, plants, and just trying to compare them. So remember, a plant is really important. They go through the process of photosynthesis in order to generate energy for themselves. Well, with a tree, because the tree is so large, it actually exhibits a secondary growth, which is that production of the woody tissue. So that wood actually allows for more support, as well as when we look at that tree, at the roots, there's bacterial interactions. And remember, this would be a mutualistic relationship because it allows for the bacteria to take nitrogen in a non-usable form and fix it so that the plant can actually use it in their daily life as a usable nutrient. So when we look at a plant, how do we know if our plant is a tree or what makes a plant a tree? Because a shrub and a bush technically aren't trees, but they still have that woody tissue. Well, the main thing is they're still autotrophs, so they're still producing their own energy through photosynthesis. But the difference between them and others, they have a little bit more rigid of a cell wall. They still have that large central vacuole and the chloroplast. But the key is that secondary growth. That secondary growth means that there's going to be the production of wood. The thing is, though, is that this growth doesn't just happen in one direction. It happens in all directions. So the tree actually grows from the center out, and it keeps adding more and more growth each year of its life, which is kind of cool. So again, if you start at the very, very middle, and then it continues to grow and grow and grow, there are cells that are going to be produced on their outer layers. We also have vascular tissue, and what vascular tissue is if you think of vessels, or you consider us as humans, our vascular tissues are our veins, arteries, capillaries, things like that. Those all transport all of our important nutrients. So for a plant, our vascular tissue, or their vascular tissue, is the phloem and the xylem. The xylem transports water, and the phloem transports sap, or sugars. The sugars travel down from the leaves through the plant to the roots. The water travels from the roots up to the actual leaves because the leaves need that water for photosynthesis. So if you take a look here, you can actually see that there are Cambrium cells, which those are very similar to our own stem cells. And then they will actually divide and then differentiate. So similar to us, our red blood cells, white blood cells, all came from one specific stem cell. And then it differentiated and had different functions. So same deal, that Cambrium is going to differentiate into either xylem to the middle or phloem to the outer portion of the tree. So if you take a look at a tree and it's been girdled, which means they take like a rope or a saw and they cut around the outside part of the tree. They don't even have to cut all the way through it. If they disrupt that Cambrium and they kind of cut off the xylem and the phloem, then there's not going to be any transferring of water or sap, so the tree basically dies. So there's some plant hormones that I want you guys to be familiar with, and we're going to go through each of those. So auxins actually promote cell growth, and this is involved with gravitropism and phototropism, which means the response to gravity. That's why the roots grow down and the stems grow up. Phototropism is the growth towards light, so you'll see the plant actually follows the light and grows towards it during the day. Gibberellins promote stem elongation. Cytokines actually promote the cell divisions and organ differentiation. Hibiscus acid promotes seed dormancy and causes our stomata to open and close. That stomata is that small opening on the other underside of the actual leaf. That small opening will have two guard cells, so you can see 1, 2 right there. And they either swell in size, causing that hole to close up, or they shrink in size, allowing for it to open up. And that process allows for gas exchange. Ethylene is going to be the gaseous hormone that actually helps with ripening of plants. and ripening of fruits. So when we look at plants, you want to make sure that you aren't putting an apple close to a banana, because apples release a lot of ethylene, and so then the banana will turn brown quicker and faster. If you do have a banana, though, that is green and you want it to be ripened for lunch, if you put it in a Ziploc baggie, that will trap those gases and it will cause it to ripen a little bit quicker. So again, each year there is one annual ring that is added onto a tree. So again, that vascular tissue continues to grow and grow and grow, radially out, so that is out in all directions. That's what it means for it to be radial growth. Okay. Each year equals 1 year of a ring. Dendrochronology is the technical term for study of annual rings in trees. And so when we look at those rings, we can see different widths of the rings. So you can see, depending upon the type of climate, was it a good growing season or a poor growing season? You can see if there was an infection. You could see if there was a fire. We can also count back, if we know when the tree was cut down, to find different historical things that happened over the years of that plant's life. Now, annual rings aren't the only thing that we can look at in order to identify climate. You guys probably know about ice cores. We can look at how much of the ice melted and froze each year, but we can also look at sediments, and we can look at minerals and stuff like that from caves and from the corals. So again, each year is just underneath the bark. So right under the bark, that is going to be the current year's growth, okay? And then that's going to continue to grow and grow and grow. And if we look at this picture, there is 1, 2, 3, 4, 5, 6, 7, 8. So it is in its 9th year of life. So you might wonder, why is there a lighter portion and then a darker portion? Well, it has to deal with the size of the actual cells. The actual size of the early wood or spring wood are a lot larger and they're lighter in color. Whereas in the late wood or you're going to notice that the cells are a lot smaller and more denser together. So that's kind of where you see that black region here. So again, in early wood or spring wood, these are thinner walls, larger diameters. They appear lighter in color. And then late wood are the ones that are thicker walls and they are smaller in diameter. Now you might be wondering why this actually happens. Well, think about this. If this is during the spring, what happens in the spring? Hopefully you're saying there's a lot of rain. So we know that there's going to be a lot of water available, so that would make sense as to why the actual xylem are a little bit bigger, because it has more water that needs to be conducted all the way up to the leaves. Not only that, that's the early portion of the growing season where we see more growth and we see a greater production of our sugars that then eventually get stored as saps. So during the late wood, when we're looking at fall into winter, the temperatures are changing, the days are getting shorter, so there's less and less sunlight, so less sugars are going to be produced, and we start going into this mode of controlling how much water is lost and trying to be conservative. Okay, so again, if you look, the vessels in the early wood are a lot larger than in the late wood. The late wood, again, would be smaller because there's less water available. So again, another way to say early wood would be spring wood. Another way to say late wood would be autumn wood. So you might see it both ways on a paper. So again, each year we are adding on a new ring. There is a way that you can actually collect a core sample without killing the tree. You don't necessarily have to cut down a tree to see its total age, which is kind of cool. Okay. So when you think about the different rings, why some are smaller and some are larger, if you notice, a lot of these are pretty big. These ones at the end are a lot smaller. Well, that makes sense. At the earliest part of your life, do you grow more? So think about from age 0 to 18, you grow a lot, right? But from age 20 to 30, do you really grow that much? Not really. So it's very similar with a tree cell. So again, variations in the rings can be caused by a lot of different factors. It can be environmental, availability of water. There could be a pollutant in the area. There might be more competition with other species. So a lot of those changes can actually allow for us to identify the end-all climate of that time period back then. So again, here's the cores. Okay, so you can actually use this tool right here in order to extract the core without completely destroying that vascular cambrium. So that means that the xylem and phloem are still going to be intact, and you'll still see water and sap distribution. So again, you can see this. So wider rings here at the middle, that's where it's younger and it's growing quicker. Trees have to grow really fast at the get-go because they need to get taller before any other plant does, because usually they're fighting for different nutrients. Not only do they need space, but they need sunlight as well. So they gotta get as tall as they possibly can, as quick as they possibly can. So heartwood, if you take a look, heartwood versus sapwood, heartwood is the dead inner portion of the wood. So it's kind of gross because it's kind of like the waste of the tree just kind of gets short because you don't really have a tree that's gonna, go to the bathroom. So that actually gets deposited to the inside part, whereas the outermost part of the tree is actually gonna be living. So again, this discolored portion right here, that would be your heartwood at the very, very center or at the core, hence where the term heart comes from. So because of its age, that's why you see the change in the color. But not all discolored wood is considered to be heartwood. So you gotta be a little careful on that. And not all trees form heartwood. Okay, so cool thing about trees is that they can live longer than all other plants. They can get bigger than all other plants. Think of the California redwoods. Oh, those things are gigantic. Okay, they can respond to damage, disease, insects, and environmental conditions successfully, which even if there is a fire, they can survive that. It's just, again, you're gonna see limited growth. And trees are a long-term investment. So if you do plant a tree, that tree is gonna be there for a very, very long time, which is super nice. All right, guys, you have an activity to do with tree dating and practicing dendrochronology. So I want you guys to give that a try. And then if you have any time left at the end of the period, can you please use that in order to complete anything you are missing? Make sure all your stuff is submitted from Thursday, Friday, and today. And then that way we can pick back up tomorrow on Tuesday. All right, you guys have a great one. Thanks.